This video lecture will talk about developmental disorders within the nervous system, taking a look at the structures involved that can cause complications. When we take a look at the various developmental disorders, we're going to see that they, for the most part, all boil down to a structure called the neural tube. The neural tube is the embryonic central nervous system. Now, that means that during early embryonic development, when you were forming into a little ball of cells way early in the first trimester, those cells started to organize into layers. And along the posterior surface, that outer layer is going to fold a long tube underneath called the neural tube. And that's what we see in blue here. So we're looking down on the posterior surface. And as this tube forms, it's going to start sealing upward, almost like zippering up, and sealing downward, zippering down. Once it closes off those two ends, called neuropores, that's going to stimulate growth of the structures. It's going to make more cerebral spinal fluid inside, which is going to stimulate more growth of nervous tissue. The upper portion is going to grow into the cerebrum, the brainstem, and the rest of the brain. The lower portion is going to form the spinal cord and all the nerve plexes, which will direct development of your girdles and your limbs. And if something interferes with that, that's where we're going to see these neural tube defects. As an example, spina bifida. Spina bifida is when the posterior neuropore fails to close. That's the bottom hole. And so if that fails to close, then it's not going to fully form all the lower structures. And as it describes on the slide here, the amount of deficit depends on how much of the tube fails to close and how early in that first trimester. For example, the most minimal part of spina bifida is called spina bifida occulta. Remember that occulta means hidden. And so in this case, it's so completed that the nervous tissue forms the entire spinal cord and it forms the nerve plexuses, but the outlying bone that forms the protective vertebra and the sacrum don't quite finish. You end up with essentially this big soft spot, this big dimple in the butt, and that's a very minor detail. That's the most minimal part. From there, we could take a look at the spina bifida meningocele, and this is where the spinal cord is intact, but then the meninges that contain all that cerebral spinal fluid bulges out past that little opening in the vertebra down at the bottom and forms this big fluid-filled cyst almost. It's not really a cyst because it's not sealed by itself. It's still circulating fluid with the rest of the central nervous system. This big bubble has cerebral spinal fluid. This is surgically easy to fix. We can close it off. We can do some reconstructive surgery and fix that. That's not a big deal. Get a little bit more severe, we get into spina bifida meningomyelocele. Watch your spelling on this. Practice, practice, practice. In this one, we have that bubble of cerebral spinal fluid in the meninges, but we also have some of the neural tissue has reached out into that bubble of meninges. And so this can draw some of the spinal cord, possibly some of the plexuses out there, and that's going to interfere with proper wiring down below the lower limbs. The most severe form is the spina bifida myelocele. With this, you had a major portion of the bottom neural tube not close off. And so you essentially, again, depends on how much, but this could be so severe that the bottom nerve plexuses simply didn't form. You may not have nerves running down to the lower limbs, and that's going to stunt the development of bones, muscles, and blood vessels. And maybe so far as have like just tiny little flippers sticking out of your hips. You don't have pelvic structures that have finished off. You won't have all the sphincters for the urinary system or the digestive system. So there can be a lot of developmental loss there. We see two examples here. On the left, this is going to be our spina bifida meningocele. And in this one, we have this bubble full of cerebral spinal fluid, but the spinal cord and plexuses are normal. And so the lower limbs have formed normally. This one's easy to fix. Over on the right, we have a severe meningomyelocele. And this one is full of a lot of neural tissue and is not properly wired down. You can see these limbs having severe club footing and severe misalignment of nervous tissue. We're getting better and better with surgery, being able to identify this early, because remember, this is still early in the first trimester. And so if we identify early enough, there are surgeries we can perform before delivery where we go in and we can fix some of those pieces and try to let them continue growing as normally as possible. So it's an amazing thing, but this would be something that you probably would have like a family history of. So you'd know to be doing very high scan ultrasounds that early and have access to this kind of specialized obstetric surgery. Now that was down spina bifida in the posterior neuropore. Anencephaly is a result if the anterior neuropore fails to close. Anencephaly in Latin means no brain. If the anterior neuropore fails to close, you're going to have a loss of brain structures. And again, how bad depends on how much and how early in development. You see a severe example here where there's a lack of most of the cerebrum. An individual this severe probably is not going to survive past delivery. Mother's life support mechanisms will keep oxygenated blood flowing through. Her kidneys will filter out waste, and all this will survive until you disconnect, and then it'll probably not survive. It could be minimal enough that most of the brain is fully intact and may just be a large fontanelle soft spot in the skull, and we may have to do some, surgery and even possibly like, some headgear until everything forms properly. Whole spectrum of how little or how much. Hydrocephalus, oops, hydrocephalus is when you have, as the name implies, water on the brain. Cerebral spinal fluid has overdeveloped within the brain itself. Remember that on the inside of the brain, we have these hollow spaces called ventricles, and they have tissue that are constantly filtering blood to make cerebral spinal fluid to feed and cleanse the nervous tissue. So they'll do it all in the spinal cord and brain from the inside, drain it down out to that subarachnoid space on the outside, clean and drain that, and then flow back out to the venous system. If you have hydrocephalus, something has blocked off an area here. Could be trauma, could be some developmental occlusion. The ventricles won't stop making cerebral spinal fluid. They have to. They need fresh fluid. They need fresh oxygen. And this is going to fill up the inside, and that pressure of fluid is going to push this very soft and spongy brain tissue and push it against the skull. And severe enough, this could cause severe intellectual disability and a lot of damage to structures. If we detect it early enough, and if it's a situation that we can get to surgically, we could put a shunt inside. And I've done dissections on cadavers where you see a plate that was, they took out a big skull bone and they went inside and carefully put a shunt. And if they can just get past it, they're done. If they can't, then they will put a little hose that will come out into the plate and it'll go under the skin and down into the thoracic cavity. And so excess cerebral spinal fluid will simply drain down into the thoracic cavity, be reabsorbed by the pericardial membranes and the pleural membranes, and the patient can be just fine. As I said, I've seen cadavers where they've had this in place for decades and they've lived just fine. So depending on how severe and how early this happens and how quickly we catch it is a big, big factor. Microcephaly is a smaller brain, which usually is due to a smaller head due to a smaller brain. Something has caused all the brain structures to form. So this is different than anencephaly. So the cerebrum and everything else is here, but they're very stunted and very atrophied and a lot less complex than a fully formed brain that we see in our right side example. This can be due to chemical exposure. It can be due to infections like Zika virus. We'll see later this chapter. There can be other traumatic reasons. There can be a variety of reasons that can cause something like this. Be mindful of what microcephaly means. As you look through each one of these, consider what they would do in terms of affecting the central nervous system. What would be the deficits that you would expect to see in an individual with a situation like this? Look through your materials, Let me know if you have any questions. Practice building your flashcards, and good luck.